Good evening. I'm Dave Long. I'm the co-president of the Marin chapter of CNPS. I want to welcome you to our November public meeting. We're going to be talking about wetlands and wetland restoration and invasive plants tonight. And we have two experts from the Coastal Conservancy Invasive Spartina project. Drew Kerr is a treatment program manager with the project, and he has, before that, he was in Seattle as a aquatic noxious weed specialist with Kings County Department of Natural Resources, and he has degrees from the University of Michigan and a certificate in wetland science and management from the University of Washington. Simon Gunner is a senior botanist at Olafson Environmental, which works with the Spartina project, and has an MA in biology from Cornell, and has interned at Harvard and the New York Botanical Garden in plant systematic laboratory. He develops cordgrass ID programs and maintains the geodatabase and assists in project design. So it's with pleasure that I turn this over first to Drew, and then Simon. So,
hi everyone. I actually will start.
Oh, I'm sorry. I'm
gonna try to start at least. I'm gonna start my share. All right. How does that look?
Good. Looks good.
Well, thank you everyone for joining us today to learn about our native and invasive Spartina species in the SF Bay estuary. I'm going to talk about the history of bay conservation broadly and how the invasive Spartina project fits in. And then my coworker Drew is going to talk about treating invasive Spartina species and our restoration work. I will say, going into this, our company pronounces the word as Spartina. Spartina is also correct. If you hear me use Spartina, though, it's just because it's how we say it around the office. Bear with me. But we're excited to be here and to share our work, and we're happy to take questions at the end about the work we do. So our work happens in tidally influenced wetlands where freshwater inputs meet the ocean's salty waters. Tidal salt marshes are very important to the health of the San Francisco Bay and to us humans who live and work and play on the edges. Tidal marshes are sandwiched between the marine and terrestrial ecosystems. Tidal marshes are essential for, are essentially the upper vegetated portion of mudflats that ring our bay. And they're highly productive ecosystems rich in biodiversity. They provide critical habitat for numerous aquatic and terrestrial critters who feed and find shelter and breed in salt marshes. They also provide ecosystem services of significant commercial and recreational value to people. They are important carbon sinks, helpful in combating climate change, and they protect our water quality by filtering contaminants from urban runoff. I'll say for context, globally, about 25 to 50% of tidal salt marshes have been lost to human development. Oops, excuse me. In the Bay Area, about 85% of tidal wetlands in the SF Bay estuary have been lost due to human development. These maps were created by SFEI to show the extent and the loss of our tidal wetlands in the Bay. You can see the hotter green in the now image showing our remaining tidal wetlands, with the more neutral green showing restored baylands and dark green showing proposed restoration, but still not quite at our historical levels. You can see a lot of the land had been diked and filled for agriculture and residential and commercial development, and especially in the South Bay, levied for commercial salt production. The SF Bay estuary still contains the largest remaining expanse of tidal salt marshes in the Western United States. And it is the second largest estuary in the entire country. It is important wintering habitat for waterfowl and shorebirds, and very important stopover area for more than 1 million migratory birds each year. So all this historical development means that our remaining tidal marshes have many important tasks on their shrinking shoulders. Where they are found, they do still protect urban areas from flooding and storm surge. They reduce erosion. They filter pollutants from the water and sequester carbon. And they provide an open space for recreation. But they also provide space for increasingly rare species suited to the salt marshes, including the salt marsh harvest mouse, the California ridgeways rail, the California black rail, as well as California sea blight, Swida californica, and soft salty bird's beak, Chloropyron male, subspecies male. So this slide is just a tiny window into the San Francisco Bay conservation history. Notably, in 1961, 3 women who lived in the East Bay founded the Save the Bay to fight the Reber Plan. I don't know how many people are familiar with the Reber Plan, but it was this crazy idea to fill in about 20,000 acres of bay. And the Save the Bay group, their vision helped turn the bay from this place to develop and a dumping ground into the treasured open space we see it as today. Over time, with additional legislation, wetlands projects, and the establishment of agencies like the California Coastal Conservancy, they're ushered in a new age of bayland restoration. So important to our story, Spartina alterniflora and East Coast cordgrass was introduced through a restoration project in the 1970s. Its aggressive spread, however, was noticed by biologists and researchers like those at UC Davis in the 80s and 90s, and their efforts resulted in the formation of the Invasive Spartina project into the year 2000. And while individual landowners like the US Fish and Wildlife Service and the East Bay Regional Parks District worked to control hybrid Spartina on their lands, they realized quickly that a rhizomatous plant like this can spread on the tide and needed a regionally coordinated effort to maintain native robust habitat throughout the bay and achieve the goals of restoring a healthy bay. So we got the State Coastal Conservancy and the US Fish and Wildlife Service are tasked currently with protecting salt marsh species and are co-leads of the Invasive Spartina project. And the California Invasive Plant Council, Cal-IPSI, and OEI, Olufsen Environmental, manage and conduct the work on the ground. In addition to these 4 organizations, we have over 200 partners around the estuary. As you can imagine, imagine the bay, any bay edge with any jurisdiction, agency, or landowner, with any lands that touch the bay waters. They may have habitat where Spartina can grow. They may have marshes that they manage. And these are cities, counties, local and state and federal agencies, water districts, mosquito abatement districts, and many private landowners as well. We work with all these partners to achieve our goal. I wanted to introduce you, in case you haven't met, Spartina foliosa. It's the only native Spartina species in the estuary. The plant is also known as Spirobolus foliosus due to a recent taxonomic revision, but we'll just stick with Spartina foliosa for this presentation. And you may even hear me call it endearingly Foley for short. But it is a perennial halophytic, so salt-loving plant in the Poaceae family. It's rhizomatous and can spread using their lateral underground tillers and shoots. And through clonal growth, it can establish itself on upper mudflat. Its geographic distribution is limited to California and the Baja Peninsula, Baja California of Mexico. It is a foundational species in low marsh and grows in a narrow elevational range, as you can see on the diagram. It stays in its lane, if you will. If you can look on the diagram into that kind of one section of marsh, low marsh. And it is also the only low marsh species that grows in this higher salinity zone in the bay. So it provides 100% of habitat cover there for wildlife. Also, fortunately, more than 99% of the cordgrass in the estuary is our native Spartina foliosa today. But there are other players in the Spartina genus. So there are a number of problem Spartina species, including English cordgrass, Spartina anglica, which was misidentified as Spartina densiflora when it was used in a restoration project, and Spartina densiflora, which is another problem species we have today, Chilean cordgrass. Both are now found primarily exclusively in the Corte Madera Creek watershed, but there'll be more on them later from my coworker, Drew. We also have salt meadow cordgrass, Spartina patens, and that's only found in Southampton Marsh in Benicia State Recreation Area. And all three of those are close to eradication. But I'm going to focus instead on the bigger problem species or taxa, smooth cordgrass, Spartina alterniflora. It is a plant native to the Atlantic and Gulf coasts from North to South America. And it's hybrids with our native Spartina foliosa. This plant and its hybrids are rhizomatous perennials found throughout the bay in salt marshes, on mudflats, on riprap, in waterways. The California Invasive Plant Council, Calipsy, rates it as high. So of all the species, Spartina alterniflora and its hybrids have the largest impact, I would definitely say. But what happened to start this invasion? Well, in the 70s, it was introduced by the Army Corps of Engineers to stabilize dredge spoils for tidal marsh restoration. Best intentions, unintentional consequences. We have a better understanding now of what happens when you introduce a novel species to an established ecosystem. The hybridization that happened, alterniflora with our native foliosa, likely really happened in the 80s and was fully identified in the mid-90s when folks started studying this new species taxa using genetics. So scientists at UC Davis described the backcross hybridization that was happening over multiple generations by that point. A backcrossing that produced highly fertile hybrid swarm, we call it. So this is a swarm of hybrids with many different forms and morphologies. And these differences allow the hybrid to exploit all tidal niches using different traits and strengths. So the hybridization between the species from different coasts resulted in plants with classic hybrid vigor. That's where offspring have more robust characters than either parent. So we normally think about hybrid Spartina as characterized by having greater cover in its clone. So increasing competition and exclusion of all other native marsh plants around it through its cover. It has a greater height, which allows it to establish itself at lower tidal elevations and keep its flowers above the high tide mark. And it has a greater inflorescence size, which allows for pollen swamping of adjacent native cordgrass, as well as having a greater seed production to produce more seeds. So this greater tidal range that is also afforded it is shown in the diagrams comparing the foliosa tidal range on the left to the alterniflora and hybrids range on the right. You can see that the hybrid does not stay in its own lane. Here's a really cool example of San Lorenzo Creek mouth in the East Bay, part of Roberts Landing in San Leandro. You can notice the comparison from 1986 to 2004. San Lorenzo Creek mouth has had a lot more expansion and accretion out into the mudflats, so much so that Spartina clogged the waterway trying to flow out via this channel. It's considered an ecosystem engineer, and you can even see there's a tiny clone out on the mudflat at the very top of the 2004 picture that would have that clone would have undoubtedly merged with the mainland and converted all of that space to a hybrid monoculture instead of leaving it as a valuable mudflat habitat were it not for the work of the Invasive Spartina project. Other problems though, hybrid Spartina has thick roots and can produce so much underground biomass that it excludes invertebrates, which are the food base for larger vertebrates like birds and fish, also crabs. If left unchecked, hybrid Spartina could cause the extinction of our native Pacific cordgrass by outcompeting and replacing it through pollen swamping. It does reduce the water flow and control channels and increases mosquitoes by creating mosquito breeding ponds. So that only leads to an increased risk of diseases such as West Nile virus. And with invasive Spartina on the loose, large-scale restoration projects are often immediately colonized and dominated by an invasive species, hybrid Spartina. So luckily, the Invasive Spartina project has worked hard to identify clones of invasive Spartina and hybrids and keep them from dominating marshes such as this one. Maybe if you look, you can spot two biologists in the picture looking for hybrid Spartina. It's a lot of area to cover and inventory, but the inventory program of the ISP works hard to cover all tidal marshes in the bay. It's not the only program we're going to be talking about today, but beyond the inventory program, there's also a treatment program as part of the ISP and a restoration program with the goal of enhancing tidal wetlands for species like the California Ridgway's rail. Drew will be talking more about these programs later. But we do have an inventory monitoring program that allows us to focus and extensively inventory and map. So this has a lot of benefits by having this kind of focused program. We send out biologists ahead of time, ahead of treatment, so that treatment crews can save time by only revisiting places in the marsh where there are features, where there are clones of invasive Spartina that have been mapped on a map. We have a tablet computer with maps on it that help us navigate. And this guided effort with a inventory program leading the way reduces impacts to sensitive habitat. Biologists can lead crews to map features, only freeing up time to thoroughly identify Spartina ahead of time, taking some time that they may need to carefully and accurately identify the invasive Spartina. And we have, I'll be talking more about the tools we use for identification, which I think are really fun. I will say that the ISP also does extensive genetic testing each year. About 400 samples of plants are taken each year to identify hybrids from the native Spartina foliosa in order to preserve it. But on our maps, we also definitely include zones outlined with special status plant species. Here's one showing chloropyron in Pinole at Whitell Marsh. We definitely keep these up to avoid trampling of any rare, endangered, or listed species. I have to say, Marin County has beautiful salt marsh plant diversity. Here is a picture showing 6 salt marsh species co-occurring in Corte Madera Ecological Reserve with Mount Tam in the background. The ISP monitors all marshes, though, for changes to plant composition in the salt marshes, and has been keeping invasive species from taking over where at all possible. I have a list of species. I don't know if you're going to quiz me later, but I'm happy to take that quiz. If you're familiar with Blackie's Pasture, a marsh, a park in Marin, there have been plantings of Sueta californica, a listed species, planted by researcher Kathy Boyer at San Francisco State University. If it hadn't been for the work of the ISP to remove the invasive hybrid Spartina, there would not have been any space for Kathy Boyer's lab to pilot plant this endangered species here. But back to identification of Spartina, grass identification. Everyone loves it. Sometimes can be tricky, but in order to protect all of not only the Spartina that's native, but all of our native plants, we need to be good at identifying invasive cordgrass. Hybrid Spartina can grow much bigger than our native Spartina, but differences are only really noticeable when the above ground stems, leaves, and flowers have had time to emerge. Right? Staff are trained in grass terminology. We even look at collars and auricles. And all of this identification is in a give-take play with our genetics program to confirm exemplary patches of Spartina. So you come across a patch in the marsh, and you, as a biologist working for the ISP, wonder, is this invasive hybrid Spartina or not? Is it the native Spartina foliosa? We examine dozens of morphological characters as quickly as possible and make our call. If plant ID seems too uncertain, staff can and will take a sample to run a genetic test. We send plant tissue to collaborators down in Los Angeles. They look at 15 microsatellites in the genetic code. So these are sections of DNA with particularly useful characteristics in order to identify the plant with high accuracy as being native or invasive hybrid. These 15 microsatellites are like 15 distinct characters, such as leaf length or shoot color, but they're only visible to a genetic analyzer. They tell us basically if the plant is foliar or not foliar. The data, though, are very clear. but not easy to come by. There is a 3-week turnaround or so. We use genetic sampling for a few different reasons, just to identify an uncertain patch, to confirm or figure out if there's a new invasion at a site that is newly opened up. Also just to establish some sure thing, definitely native, definitely hybrid baselines for comparison for other clones. And absolutely to confirm whenever we use native Spartina foliosa for restoration, that it is pure native Spartina foliosa. But it would be so nice, I thought, personally, to have a buddy in the field for when you're not sure of your ID skills when looking at a plant. For my coworkers, I developed a program using the statistical analysis, linear discriminant analysis, kind of put it into a computer program. We call her Linda for short, linear discriminant analysis. So Linda is always our buddy out there when we want real-time immediate answers. We use Linda, who has drawn from many past genetic samples as reference. And she is right, I would say about 80% of the time, looking at plants that are so heavily back-crossed, they kind of are, it's very unclear as to whether the plant is invasive or a native plant. But she's 100% correct on the more obvious plants that you can see, like in this photo showing invasive Spartina on the left, overwhelming native Spartina on the right. To support Linda, we actually have to provide her with information that our eyes can see. We find a flowering stem that, let's say, we're interested in. We use a measuring tape and calipers to measure all these parts along the plant. There's a diagram here showing some of them by color, where you would measure from and to the whole length of the plant to just the width of the stem or the leaf. And we compare the colors that are showing on the plant, and we plug them into our computer program, like in these boxes here that I have for my staff. They put in the numbers that they get, their measurements, their colors, and then they ask Linda what she thinks, and she'll say what she thinks it is. Sometimes she says it's hybrid, sometimes she says it's not. But Linda's brain is full of great information. You can see on the left, this is her understanding of the distribution of how many nodes plants have, Spartina plants have, based on whether they're the native Spartina foliosa or the invasive hybrid. You can see that the trend is that with the fewer nodes on a plant, the more likely that plant is our native foliosa. And the more nodes the plant has, the more likely it is an invasive hybrid. She uses this information from node number and lots of other characters, including even characters like leaf width, which to our eyes might not seem very clear, but luckily she's a computer and she's very smart. And even this can provide some information to her when she makes her call. Linda, the librarian, she knows all this stuff, and maybe if we don't want her to give us a holistic just call on whether a plant is foliar or invasive, she'll just let us know about the node number, about how many leaves the plant has specifically, trait by trait. And we might notice that some of the things we're looking at, some of the characters of the plant we're looking at are typical for our native foliosa. Some of the characters we're looking at may be rarely found, but still found in our foliosa. And some of them are not yet found in our foliosa in all of our sampling, and are therefore very likely to be the invasive hybrid. numbers, all this data has been really valuable, not only for us, these are data that go beyond the ranges reported in the Jepson manual for our native Spartina foliosa. And luckily, Linda is also good at knowing where you are in the bay and can give you more accurate calls based on the general region that you're finding your plants in. Because depending where you are, Spartina foliosa, our native, has tremendous diversity morphologically. There are regional differences. You might see in the North Bay, I like to tell my coworkers to, be on the lookout, because native cordgrass in the North Bay does tend to have longer inflorescences for how big it gets compared to other regions of the bay. And there are certainly elevational differences. Plant height is definitely related to inundation amounts, how much tide the plant is experiencing at a given site. But there are many differences that take a lot of different brains working on this. So I'm going to definitely stop now and give the attention over to Drew, who is another one of the many diverse and interesting people who are working on this project. So to go on, once you've discovered what invasive Spartina is out there, we're going to talk now about how we treat it. So I'm going to give it to Drew. Let's see if I can do this somewhat seamlessly. Thank you, guys.
Beautiful job, Simon.
Thank you.
That was an excellent background on the importance of our tidal marshes and the threats posed by the non-native Spartina, and all of the hard work you've done to give our staff tools, buddies to consult while in the marsh. It can be lonely out there. You have to have a buddy with you to help you make some of these tough decisions. So let me see if I can properly do this. So you seeing the right screen?
Looks good.
Okay, great. Okay, so now we're going to transition to how we use all of this inventory monitoring data that Simon has shared to manage the infestation and reduce impacts from non-native Spartina to the tidal marshes and mudflats of San Francisco Bay. So our work must be conducted, our work for the ISP treatment program must be conducted during the active growing season of the plant, especially the work on our hybrid Spartina alternate flora, the one that Simon mostly focused on, that's a disproportionate impacts around the bay of the species that he highlighted. It needs to be treated during the active growing season because we rely on herbicide due to its rhizomatous nature as the main primary tool for managing the species. However, we not only are constrained to the active growing season, we're also constrained to very narrow windows related to the endangered ridgeways rail. And most of our work has to occur at or after the end of the breeding season of the rail, end of the summer, early autumn. So we have to fit in all of the work around the entire estuary before the plant senesce. So it can be quite hectic. As Simon highlighted, we're talking about tens of thousands of acres of tidal marsh and mudflats. Simon also highlighted how the biologists conduct inventory surveys and mapping first, so the treatment crews can focus just on where they need to go. That reduces the disturbance to the tidal marsh, the denizens of the marsh, and also enhances worker health and safety out there. And we've relied on airboats quite a bit to allow us to work at a low or receding tide where standard boat propellers would not be able to access. And this actually shows one of the first days we were even using an airboat out in the marsh in 2008 at San Pablo Marsh, actually across the bay from Marin. So let's see. Yep. Okay, I'm gonna have to use the wheel. Thanks, David, for the tip on that one. So here we go. So we have now it's not showing my notes though, unfortunately. Interesting. That's a little bit not exactly the way I wanted it. Hang on a second. I'm going to take. It's funny because I just did a presentation for Calypsy and There's no issue. Let's go with this guy, share this. And then is it showing the presenters? Yep. And then if we go like that, now is it showing the right one?
Yep.
Okay, that's better. Okay, great. So the high-speed treatment program, we treat the larger infestations are treated by hauling hose out from airboats or trucks out into the marsh to apply the herbicide. And we approach from the mudflat edge to reduce disturbance to wildlife that's associated with the denizens of the marsh plain, and having to not cross as many channels, not cross the marsh plain and create the disturbance for ridgeways, rail, salt marsh harvest mouse, and other species. But as you can see by the footprints there in the mud, it can be quite soft and hectic getting out of the airboat to conduct this work, and quite muddy. However, the majority of the sites are simply treated by backpack sprayer. This graph shows the tremendous success that we've realized through our efforts over the years, starting back in 2005, when we had about 805 net acres. And by 2021, we've reduced that to 22.5 net acres around the bay, which represents a 97% reduction estuary wide. Now, in reality, that's net acres where we kind of reduce the spacing between the plants. Treatment acres would have represented thousands of impacted acres around the bay, especially back in the early days. This shows a series of kind of heat maps that show the infestation distribution and gives you the visual extent of where hybrid Spartina has been found in the estuary over the course of the project. Dark green color in these shows no hybrid, and red more than 1 net acre. So I know they're relatively small, but you can see in 2005 we have so much red, especially in the South Bay, and we start to move more towards orange and yellows in 2010, and how much of the estuary is in green in the South Bay by 2015. And then as we continue on into the 2020 map on the right, where we just have some moderate infestation still in the South Bay, and then also in some of the areas just north of the Bay Bridge area. We went from about 50,000 to 60,000 and now 70,000 acres around the bay that we survey. And that's primarily due to the opening up and breaching of salt ponds that acquired from Cargill. Simon mentioned this briefly. So we have a lot of restoration going on in the bay, and that is wonderful. And it's exactly what we want to see, but it expands greatly the footprint we need to monitor each year. And if we find something there, then we need to manage it as well. I think this is my favorite way to describe the progress that we've made, though. This table and the numbers associated with it. The larger overall picture is wonderful, but I think that it's really interesting to see that as of 2021, 157 of our 220 Spartina sites now contain under 10 square meters of invasive Spartina. With all those sites together, all 157 just representing a total of 195 square meters of net cover. Within that 220 sites, we have 57 sites at zero detection, many of those for 3 years or more. And that's kind of a standard of a local eradication used broadly in the weed world of achieving 3 years of zero detection. Of course, you could have reinfestation, and then that's why we're going back to these sites and monitoring, especially when you have hybrid Spartina proximate to it. And these sites, of course, encompass tens of thousands of acres of marsh and, very importantly, mudflat. We are explicitly trying to protect and restore mudflat to its unvegetated state to protect that shorebird foraging habitat. That's often a misunderstanding of our project when you have these large green meadows that were dominated by an invasive plant, and we return it to mudflat. That's actually purely was the intended goal. So I want to focus a bit on our Spartina densiflora integrated vegetation management strategy, because it's a very interesting, very, very complex one. And it really is very pertinent to the Marin audience, because most of the Spartina densiflora in the bay was found in Corte Madera Creek and eastern Marin County, as well as some over in Point Pinole, but almost all exclusively in the North Bay. Spartina densiflora was introduced from Chile to Humboldt Bay. It was brought up in the timber trade in the 19th century. It was used as dry ballast, so the ships would be leaving Chile. They would load up with a bunch of the marsh cordgrass right next to their boats, and then they would dump it off in Humboldt when they got there and load up with redwood logs and head back to South America. Unfortunately, it was mistakenly identified as a form of the native back in the 1970s when Creekside Park, which is now called Hal Brown Park, was undergoing restoration. And so it was, the densiflora was planted at that time, even though it's a bunch grass and looks nothing truly like Spartina folios or folia, as Simon emphasized to a botanist. Unfortunately, it was introduced there, and that started the infestation. Those are some big plants there, as well as you can see the continuous fringe of Spartina densiflora along Coromandel Creek back in the early days of our project. The fortunate thing about Spartina densiflora is because it's not rhizomatous like Spartina alterniflora or the hybrid with Spartina alterniflora, individual Spartina densiflora can be dug out effectively without stimulating vegetative growth. However, large-scale removal can be damaging to the marsh surface. You can think of accreted marsh plain as an old growth feature that took many years to develop. So we want to minimize disturbance to it, since micro-elevational differences dictate the plant community in tidal marshes. So we developed an IPM strategy, or an integrated pest management strategy, with a imazapyr herbicide to reduce the impact, but relied on a variety of methods to achieve our goals. Sandy Goldman, longtime president of Friends of Coromandel Creek, contacted literally hundreds of individual landowners to gain access permission for inventory and treatment over the years. Some final holdouts, folks that weren't interested in participating in it originally, required contact from the Marin County Ag Commissioner and education on the state noxious weed law, but eventually all partnered with us and without any need for enforcement actions. So I call the marsh, after we had treated some of it with herbicide, the Spartina densiflora marsh, the marsh of the living dead. And unlike our friend Spartina alterniflora hybrid, which is the bulk of the infestation around the bay that Simon focused on, which is very effectively treated with a imazapyr herbicide, this bunch grass, unfortunately, is not as effectively treated. So established stands of Spartina densiflora can display this kind of yellow, green, gray, half-dead appearance about 1 year after herbicide treatment. And they're not healthy enough to translocate another herbicide application. Plus, this necromass, as we call it, or this standing dead biomass, is very persistent. So we developed a strategy where we experimented with mowing the previously herbicide-treated Spartina densiflora on the mid-marsh, the pickleweed plain, the mid-marsh pickleweed plain, and that removed the dead or partially dead above-ground biomass and allowed us for best assessments of the current plant status. Are these plants dead? Are they alive? Are they going to resprout? And that mowing, as most folks are aware, can add stress to invasive root reserves. So it did actually eliminate some of the plants too, between the combination of the two methods of treatment. And you can see those accreted mounds formed by the mature Spartina densiflora plants that had been living there for decades. Then we went in with a imazapyr herbicide, and we did a post-mowing imazapyr application to the Spartina densiflora. And this was done with the intention of arresting the development of the plants and stopping the seed production and dispersal. We estimate the seed bank to be about at least 5 years. So we want to stop that seed production. And so we implemented this on the regrowth, the season after mowing. You can see a lot of smaller plants that the gentleman there is treating. So that mowing greatly reduced the amount of herbicide needed due to less above ground biomass. And not having to dig all these plants preserved the integrity of the marsh plain that I just told you about. We want to preserve that because of the micro elevational differences that dictate the plant community. So only a fraction of these plants now needed to be dug, as opposed to coming in with some large scale, very disturbing excavation tool. The higher disturbance activities, in other words, disturbance to Ridgway's rail, were conducted outside of Ridgway's rail breeding season, when we would bring in crews to do the digging or do the mowing, where we could come in about an hour and do a very quiet, measured herbicide application, and at least stop that seed production, kill a few plants, and contribute to this integrated pest management strategy. So it was very effective, and the strategy, the combination of the methods was very effective. So that's what IPM is, you know, understanding the biology of the plant, responding to the biology of that plant by looking at what are the best tools in our toolbox, in our armamentarium to respond to this situation and get the native plant community healthy again, so that it can resist invasion and provide its habitat services, functions, and services for the ecosystem. So this was turned back to lush pickleweed and Spartina foliosa colonizing the main channel of Creekside Park, where you can see the exact same area where I was mowing a couple years prior. You can see a meadow of the Spartina densiflora here being treated with herbicide in the early days before mowing began in 2009. And you can see a couple years later, this is actually 4 years later, you have the results are, you know, tall, dense pickleweed and saltgrass. This is passive restoration too. This is revegetation that happened with the native plant community that was already on site. Just removing that invasive over the course of years with those multiple methods allowed the native plant community to burgeon again. And we, you know, have this diverse assemblage of plants. And this is, again, the annual management peer treatment in June, mowing to the substrate in around October, and then spot digging the plants that we need to remove over several years as well. And you can also see caged transition zone plantings from the Friends of Coromandel Creek over there in the top part of the photo. So our current Spartina densiflora eradication methodology is to all historical sites are surveyed twice annually, once in early June, where the flower stalks, if they're present, can help with detection. The second is in January, February, when I say the native marsh is sleeping. Most of the native plants, including pickleweed, have, you know, senesced back to their perennial roots, and the dark green of the Spartina densiflora stands out, and we can detect it even as very small individuals, as you'll see in a second. All plants are manually removed at this point in the eradication. We started moving us away from herbicide use for this species with our IPM back in 2009, and we have not had to rely on herbicide at all for densiflora since 2012. At this point, throughout the entire estuary in 2021, we found 2.1 square meters, a 99.9% reduction. And you can see the tiny little individuals that our biologists have to go out and find when they do their twice annual surveys. And so it's very laborious. When we have the data on our GPS units, when we find a concentration of old features where we spend a lot of time in those areas, walking at them from one angle and another angle, if the sun is bright, if the sun is harsh, giving ourselves the opportunity to find these small individuals, removing them manually. So we had 27 seedlings found throughout the estuary in 2021, and that's down from 65 in 2020. So we're making great progress, but you can see what full eradication, that term gets thrown around a lot, eradication, is a very, very high bar and requires long-term stewardship. We think that it's about a 5-year seed viability for this species, but it could be more. So now I'm going to transition to the ISP's restoration program and our habitat enhancements, which are centered on Ridgway's rail. I'll be showing some slides from our restoration program manager, Jeannie Hammond, who some of you may have seen give a presentation at the CNPS conference in San Jose a couple weeks ago. So the hybrid Spartina, the main one that moving back away from the densiflora, which was smaller in scale and had impacts, but not the level, it can't grow to the mudflat level like the hybrid Spartina alternate flora that Simon focused on so much. The hybrid Spartina invasion not only extirpated the native cordgrass, the native Spartina foliosa, but it also impeded the establishment of a diverse assemblage of native plants at early restoration sites around the bay from around the 1980s on. And while it also overwhelmed Grindelia and other natives at historic marshes. So that invasion greatly limited the potential of native propagule sources to exist within big sectors of the bay. There was no longer a really good seed source for Grindelia or even for the native Spartina anymore. So that necessitated active planting restoration in addition to the cordgrass treatment that we did to get to free up these marshes from the threats posed by the invasive plant. We focused on key components of rail habitat, cover from predators for foraging, roosting, nesting, and high tide refuge. And this is supposed to be rapid active enhancement where we do the passive revegetation of the plant community will happen with time, but we're trying to help keep pace with sea level rise. And so we need to speed up the establishment of vegetation through active planting efforts. So over the 11 years of our project, of the restoration portion of our project, the map shows the 40+ sites that we have implemented habitat enhancements. As previously mentioned, these are focused really on the areas with the greatest impact from the hybrid invasion and its subsequent treatment, which is the South Bay, closer to the introduction areas where it had decades to fester and reach the massive monocultures impacting hundreds of acres of native Spartina. The blue in the map shows the plantings primarily planted with Pacific cordgrass and marsh gum plant, Grindelia stricta. And the orange shows where high tide refuge island features were constructed, 82 of them at 16 sites. I'm going to get into that in a second. But we planted over 550,000 plants in those 11 years at those 40 sites. And many of these are very hard to access, require airboat access, and all this is done kind of on our off season from monitoring and treatment over the winter, starting in December through February, March. What do we plant? Again, Pacific cordgrass planted in, this is that same marsh zonation diagram you saw previously that Simon was showing us. We want the Pacific cordgrass to establish in that low marsh area and just up into the middle of marsh. We used the marsh gum plant is planted primarily along channels where it likes the well-drained channels, and it provides key nesting habitat for Ridgway's rail. They'll use monocots to form their nests within that marsh gum plant and then jump back down into the channels to forage. So it's very important that we have that feature, and a lot of that was not present in these marshes after the invasion. And then we've also moved into transition zone plantings in the as you move from the high marsh up into the upland area, which are rare features, unfortunately, in the bay, because we've had so much development right up to the bay edge, and so many marshes don't have very much of a high marsh transition zone, upland transition zone. So the vertical cover for Ridgway's rail at high tides is provided by a pretty small suite of plant species, and they're the ones that we focus on here. And the photos, the before and after photos really show the utter transformation of habitat quality from the reintroduction of these two native plant species over just say 4 or 5 years. Sometimes you can see the cordgrass transition from basically planting in the open mud at a new restoration site to a nice lush lawn of the native cordgrass, taking a channel bank and planting it with Grindelia. So now you have all that vertical structure that the rails can use both for nesting and for refugia, and then getting into some of the upland transition zones too, which can be critical habitat at that high tide and for refugia as well. And then once these plants are established in these marshes again, there is a propagule source present, and it can disperse within the site and find its own way passively. We don't need to continually actively enhance the habitat. Just a quick look at the kind of the planting designs for rapid enhancement, and we're really focusing on doing it at a very high density grid planting. That's also a design that's easy and fast for contractors to install. So we have the Pacific cordgrass one on the left-hand side showing the 5-plug block design for native cordgrass, and then you have the Grindelia nodes of habitat, the two plots of the larger pots, the larger circles of usually gallons, and then the smaller ones planted, usually now D16s or D40 sometimes, planted along the channel very densely so that we will, get that habitat value as quickly as possible. And the idea is that, a Ridgway's rail pair would use the channel for foraging and then pop into this node of habitat cover to escape predators or to roost or to nest. And then we repeat these nodes whenever needed to support additional habitat throughout the site. Then you have the marsh upland transition zone species, ambrosia, baccharis, disticulus, euthamia, and frankinia, the usual suspects that do a really good job at filling in and surviving in that transition zone. And I mentioned the high tide refuge islands, kind of a novel approach to giving some vertical structure to some of these marshes. The highest mortality for Ridgway's rails happens during winter extreme high tide events, especially those that coincide with winter storm events. So the Invasive Spartina project and Coastal Conservancy collaborated with H.T. Harvey and USGS to construct these small islands adjacent to tidal sloughs in marshes with Ridgway's rail. They're about 30 square feet in size, and the island is immediately adjacent to the slough, and ideally a slough channel a confluence that will facilitate rail use. So it's very interesting how this the methodology for this. We first remove the marsh plain, we call them a sod, basically the sod of pickleweed plain, and we stack that to the side over the whole footprint of that, while the base of the island is constructed with the adjacent slough channel substrate. So there's no need for costly soil importing. We're using the substrate of the channel, so we're not damaging the marsh plain. We're using it down in the consolidated sediments down in the channel itself. Then on top of that, we put the sod back on top of the mound and plant that with about 60 grindelia plants, gum plants. And yeah, so we kind of strike the balance between an adequately high elevation to substantially increase refugia for the rail once the grindelia matures, without promoting upland weed invasion. So we don't want to create an upland weed node within our marshes. After about 1 or 2 years of settlement, about half a foot of settling happened. So the initial mounds were planted a little too low, and we learned the lesson and planted them a little higher. We actually, Cordgrass Ecological Reserve is actually one of the places where these have been constructed. You can see that at a high tide right next to a channel there. You can see that structure and refugia for the rail. And at Bear Island, B2 Northeast, where Simon and I were today, you can see a really beautiful island that's matured, and you can see a couple in the distance. They really stand out on the marsh plain as distinctive features. I'm going to move to some of our other restoration, some really beautiful examples of the restoration, the active restoration. And this one is from Eden Landing Ecological Reserve. This is an early restoration of a former salt pond. And I mentioned the South Bay Salt Ponds project. And, you know, we're trying to, there's the ambitious kind of wetland community effort around the bay is trying to restore about 15,000 acres of salt ponds purchased from Cargill through state, federal, and local and private funding sources. And that's a, you know, it's like a 50-year project, about $1 billion project over 50 years. This is an example, a good example of the kind of the clean slate early phase of the project. This actually was a marsh restored in 2005, had a small amount of invasive Spartina. We got that managed down to a very low level. And this shot is in 2011, where you see these open, unvegetated mudflat. So it's at the right elevation for plant colonization, but hasn't really had any yet. So we started the cordgrass plantings in 2012, you'll see in a second. And since we're very far from a local source of native cordgrass, I mentioned how the native cordgrass had been extirpated by the hybrid swarm. There was no local source, and that's why we had to kind of come in and do this active replanting of this area, even though this was a brand new breach. So we wanted to jumpstart the trajectory of the site and take it from this open mud plain to potential habitat for Ridgway's rail. So you can see on the right-hand side, all the nodes of green. Green are the active plantings of the native Spartina foliosa from years 1 through 6. And you can see the before and after shots. That's the stark difference of all those footprints in the soft mud, and then the cordgrass establishment in just a few years after that. And this, you know, this really jump-started the trajectory of this restoration site to becoming a native, healthy, mature marsh. And this is a shot I took with a drone in 2019. This is how much it changed by us introducing that and creating like the surface roughness, so that as the tides bring in more propagules, they'll drop out, and the pickleweed and other native plants will start to colonize, as well as more native cordgrass will start to move around throughout the site. And the really exciting thing about this is there was the first rail detected was in 2018, just a single rail. You know, you saw what it looked like in 2011, just a mud flat, just a mud plain is what I call it. And by 2021, there were 13 Ridgway's rail, an endangered bird, detected in this marsh. So we really were able to jumpstart and accelerate the development of this into quality habitat. And that photo is from 2019, and the vegetation has expanded quite a bit beyond that. So, and then another time series that's an interesting one. I wish we had more of these. When the Spartina project started, I started with the Spartina project in 2005. When it started, we had drones. Oh my gosh, the beautiful time series we would have had. But we did do a lot of helicopter work back in the day, and so we have some good helicopter shots. But this is probably one of the best examples showing the infestation getting worse of the hybrid, and then how much it's turned a corner and the transformation of the site. So you see in the picture there, in 2003, you see a mudflat at on the Alameda Flood Control Channel. This is, by the way, where Spartina alterniflora was introduced to the bay in the 1970s by the Army Corps of Engineers in their experimentation with dredge spoils stabilization. So you can see quite a few clones of the invasive Spartina already spraying across the mudflat, but a significant amount of mudflats still present in 2003. Then in 2005, just 2 years later, I mean, you're talking about, that's like a half mile of bend of Alameda Flood Control Channel, just covered with all these coalesced clones that are forming a meadow of 6 foot, 7 foot tall cordgrass, excluding all that important mudflat habitat that we've already discussed. And then after we did, quite a bit of control, the treatment that was required to get Alameda Flood Control Channel under control was extensive over many years. We had well over 100 acres of solid invasive Spartina at that site when we started treating there in 2005. Then we were able to come in and plant the native cordgrass, and you can see just the beautiful band of native cordgrass in exactly where it belongs, not down throughout the mudflat, but a nice narrow band at the lower elevation, and the marsh zonation shots that we've shown, and the pickleweed plain beyond it, just exactly the kind of habitat that we're looking for, basically transformed from the early days. And just to finish up here, we have this South Bay Salt Ponds project that I mentioned that's, very ambitious and over many, many acres. You have the Eden Landing Complex, the Alviso Complex, Ravenswood Phase 2 is opening up now. And, this shows a breach that I was lucky enough to actually witness in 2008 at Eden Landing. A lot of these sites, we are, hoping for the restoration to be passive. This is, where we don't have to come in and plant these sites. They're approximate to good native seed sources for all the diverse assemblage of native plants, native Spartina foliosa, Grundilia stricta, Jaumea, all the other natives. And this just shows how successful this can be. So this is Pond A6 down in Alviso area. This is all passive revegetation. It was breached in 2010, so this is about 11 years in. And it's really interesting where, it'll be a mud plain for many, many years with little pockets of vegetation. Then all of a sudden it will round a corner, and you'll start to really have this widespread vegetation establishment. And that's happened here at this site in the past couple of years. 3 years ago, you would not have seen nearly this much. So you have annual pickleweed, perennial pickleweed, native Spartina. And unfortunately, this is when these marshes have very little biotic resistance to invasion. They're in the early stages of their development. And if the hybrid cordgrass comes in, the hybrid Spartina comes into these sites at this point, it can just run roughshod over it. And really, basically it's like mud. And so it can just take off and establish and then exclude other native plants. So we want to be the good stewards of these marshes and allow this passive vegetation establishment to occur under our watchful eye. So our biologists are going out there, the OEI is inventorying these sites each year, surveying them, using the tools that Simon pointed out, the different tools that have been developed to see whether we have any hybrid Spartina here, and then we can quickly respond to that and under this watchful eye and allow these systems to flourish on their own and find their way through sea rise, et cetera, without the threat of the Spartina, the bully the bull in the china shop for sure. So, and this is Island Pond A20 down in August 2019. Again, this is breached in 2006, and you can see the beautiful establishment of native cordgrass and other vegetation throughout. So, our goal is to really do this work to provide for healthy, resilient native marshes, and where we have this kind of feedback loop where we are getting forward momentum with invasive plant management, doing restoration where needed or allowing passive revegetation to occur, and then having widespread native plant establishment and increasing rail populations that, as you saw in North Creek Marsh, 13 rails after just a short period of time of native plant establishment. So it's very important that the stewardship of these marshes, stewardship of these systems continue. And so that ongoing stewardship is very essential to maintain the health of our marshes and guard against future threats from popping up, other new invasive species or other things we aren't even aware of yet. So, and with that, I think Simon and I will be able to take questions.
Well, Drew and Simon, that was just tremendously impressive. Yeah, I had no idea that an eradication and restoration effort of that scale was going on, but more importantly, was successful. I mean, what you were portraying here is something that gives me hope that we can attack some of the other invasive problems we have, because it's very depressing to see broom expand in public lands year after year with very little happening, or maybe a lot happening, but still it's expanding. And what you've shown is that with the political and administrative will and a budget and good science, good tools, you can be effective. And that's a really important message, because it's very easy to be disheartened about invasives today.
Absolutely. You're absolutely right. And I think, the reason why a lot of folks aren't aware of this is because of the inaccessibility of a lot of these sites, right? They're not easy places for folks to just go and visit, as are maybe a park that's got a broom infestation that's flourishing. And, people aren't just taking hikes in these areas and stuff. So this is occurring kind of out of sight, out of mind. And that's also what happens with the invasion itself, right? The invasions go on for quite a while, out of sight, out of mind. And if you don't have the stewardship, if you don't have the vigilance out there to see what's going on, then these things can get out of control quick. And the best invasive species management is prevention. And after that is, early detection and rapid response. Once things get very established, it becomes costly, time consuming, and getting that political will to do it can be very challenging. So, but yes, thank you very much for the kind sentiments and kind words. It is heartening to see, and we do really love to come back to these marshes each year and see them going through these last slides, these transitions to this level of habitat. And when we're out there doing our ridgeways rail monitoring to see, to get these numbers, 13 rails in North Creek Marsh, I mean, that we were, over the moon when we heard that. So yeah, it's really neat.
Drew, could you stop sharing so I can see participants and hand raising? Great, thanks.
Yep.
We have some questions in the chat, which I'm going to get to, but I see Nona Dennis has her hand up. Nona, you can unmute and ask your question.
Yeah, sure. Okay, wonderful, wonderful story. But I do want to say that this has a 50-year history, and I think that we should give a shout out to the pioneers in the 1970s, like Phil Williams, H.T. Harvey, among others. I was involved in the early days in Muzzy Marsh and some other restorations around the bay. We've learned a lot. You obviously are showing that how much you have learned, but it is a 50-year story, and at some point, we need to give credit to the pioneers who really began to explore how this restoration was going to be done, how the invasive species were first discovered. Sandy Gouldman, Friends of Corte Creek, were among the heroes that continued working on this until they had it under control. So just a shout out to those people who have been part of the 50-year history of restoring marshes in San Francisco Bay.
Absolutely.
Thanks.
And with those people mentioned, I think it's essentially, we mentioned Phyllis Faber, who was instrumental at setting up her monitoring in Muzzy Marsh. And I'll point out that the marsh zonation diagram that Simon and I used was from Phil Williams and Associates. So we were carrying forth that, and the high tide refuge islands were a collaboration with H.T. Harvey. So that's your point is well taken, that we need to honor these the people that did a lot of work to lay the groundwork for these things to be successful, including UC Davis lab doing the genetics work for us. John Calloway at University of San Francisco, that was one of the people that really discovered the hybridization event in the 90s. Yeah, absolutely. We're on the backs of all those folks for sure.
Yeah, sure. Great. Thanks.
Hi, Sandy. Thank you so much, Sandy, for all your work. It's good.
Yeah, right. Absolutely.
That they're here. So.
Oh, she's here. Excellent. Excellent.
A question in the chat for Michael McSweeney. He asks, what about pampas grass? Is that a problem in the marsh?
Yeah, it's an upland species primarily. As we get into the upland transition zone, you can definitely find pampas grass. It will not grow within like the tidal inundation, the regularly inundated areas of the marsh. But as I mentioned, our transition zones have been restricted to these narrow little strips and stuff. So it does exist at those edges. Yes, and a big problem for sure.
Brandon, you can unmute and ask your question.
Hi, thank you, Mr. Kerr, and thank you, Mr. Gunner. My question is this: during the height of the pandemic 2020 and 2021, have you noticed an increase in the rate of the restoration of the marshlands, or any increase or decrease in the rate of the restoration during that time frame?
I would say that some marshes reached that point, like I was showing some of those slides at the end of Pond A6 or Nap Track was reaching that point. And I don't know, I don't think there was anything necessarily unique about the year 2020 that made that happen. In fact, in the lower, you'd think that these salt marsh plants would not really need rain, but we actually find that in the drought, they don't produce as much seed, and they don't expand as much. So we definitely see more of that when we have the freshwater flushing. I think that those things are more where those restoration sites were in their clock and how many years they've been present, as opposed to something within the pandemic itself, within that pandemic year. Does that get to your question, Brandon?
Gotcha. So I basically, with the pandemic, a lot of people were locked down, basically. So it was a lot less human impact to the environment, basically.
The human impacts are kind of lag effects, right? You have water quality impacts, you have impacts of these invasive species introductions, you have the impacts of us ignoring these invasive species introductions and not managing them. But luckily, we're not, people aren't walking around in these sites very much. They're sort of inaccessible to the average person. Most of them aren't out like Simon and me and Sandy, trampling in these, tromping through the areas to get to do our work. So I don't think we had a huge reduction in that would have ended up with more restoration.
Fantastic. Thank you.
Mm-hmm.
Laura Lovett is asked how resilient the new marshlands are to rising sea levels.
Well, there's, it is the kind of the jury is out a bit on that. One of the problems with our bay is that with the way that we've managed our channels, we have a lack of sediment. So we're sediment limited in our bay. So if you don't have sediment coming down from the upper watersheds to replenish the marshes, as in the natural cycles, and to accrete that sediment over time, you're going to have a hard time keeping up with sea level rise. Now, I've seen different predictions over time. I can't say that I'm up to date on all the literature right now, but things were keeping pace with sea level rise, but sea level rise was accelerating, and that was the problem. I think it was doing okay. Sea level rise is accelerating, so it's difficult. I mean, when we've also built right up to the edge of all these marshes, there's no way for them to migrate. They normally would start to migrate into these upland, into these transitions, true transition zones. But if you take away all those transition zones, where are these marshes supposed to go? So that's yeah, it's a complicated issue that we humans have made much more complicated.
Jill Templeton wants to know, what are some of the other marsh plants that are invasive, such as ice plant, salsola, soda, fennel, etc.
Ooh, yeah.
Jill was an excellent volunteer for our project early in the time when we started working with Sandy and densiflora and stuff. Used to live in the strawberry area, so I know Jill. We have a non-native limonium, a non-native sea lavender that is present in Marin County. Two of them actually, Algerian sea lavender and European sea lavender, Limonium ramosissimum and Limonium duriesculum. Salsola soda is a good example of that. We have, and that really became a lot more widespread over the past 15 years. Seen a lot, and I really think of actually some of the Corte Madera watershed and seeing that really just moving through that pickleweed plain. It kind of looks like pickleweed a little bit to the uninitiated. It's a little sort of a succulent, but it's but it's a relative Russian thistle. We have Puccinellia is a bunch grass that we just saw recently. Simon, do you want to add some species?
I have to say, a very recent invader we are actually tackling, mostly in the South Bay, is Paspalum vaginatum, and it's a turf grass that was developed for its drought resistance for use and all sorts of things, and it's so salt tolerant that it has made its way into fresher tidal salt marshes. And just by us being out there doing all the stewardship for the salt marshes, we were able to detect it relatively quite early, and we are currently mobilizing to use the same techniques that we've developed for invasive Spartina to remove that grass as well.
Yeah, and then just the knowledge of the marshes, the knowledge of how to, our airboat contractors helping us to get out to these sites, knowing how to do this stuff at low tide, extremely difficult stuff, learned by many hard days out there figuring out how to do this stuff. So that when Simon's referring to the techniques, it's the whole suite of monitoring techniques, knowledge of the tides, knowledge of where these, of how to access these sites. That, if you have entities, a team that can do that kind of thing, then you get this new information about this other invasive plant, and they can mobilize much more quickly than to just, have folks figure it out from, reinvent the wheel. So yeah, luckily salt marshes are sort of inhospitable, and so there aren't as many species, invasive species, that can tolerate it. But we've mentioned a few of them. As you get into the fennel and stuff like that, that's more in the upland as well. So other ones, Simon, anything come to?
Yeah,
I think that's pretty much, that covers the usual suspects pretty well, but
Well,
someone else is going to bring one up, I know, but
I have an additional question, and
Yeah.
and not quite sure how to ask it. You were both, invasive plant experts, specialists. You've spent a lot of years thinking about how to eradicate them and how to restore areas that have been decimated by them, and just whether you have any reflections on the invasive problem that we face in public lands, not in wetlands, but just Mount Tam, or and whether you've thought of strategies that are better or worse.
Well, I mean, you brought up an example that I have to give hats off to One Tam. I mean, who is mobilizing all these tools, and is Rachel Kiesel and a whole lot of other really talented folks have dedicated to. I think that the thing gets back to vigilance again, though. They cover many miles of trail, doing the surveys to inform. So you have the education of the teams so that they know what they're looking for. You have them getting out on the ground, looking for that at the right time of year. You want to have the tools in the toolbox to deal with these different things too. Some plants can be manually removed, some plants can be tarped. Other plants, rhizomatous plants like Spartina alterniflora, it requires an herbicide. So you don't want to rely on any single thing. You want to develop IPM strategies to deal with these. But that part of that IPM strategy is the monitoring, is the understanding of the biology of the plant, coming in at the right time. I think we have, Marin has incredible volunteer, Marin Audubon, and all the work they've done, and many others. There's so many wonderful people out there working hard to protect the ecosystems, various ones, from salt marsh edges up to One Tam to the other systems. And the Native Plant Society, just it's nurturing the appreciation of the native plants. I think that's really, if people are appreciating the native plants, they want to come and protect them from these invasive plants, and so you're doing a huge service to do that, and filling people with the joy of getting out to look at these things and looking at these beautiful plants that we love. So it's a feedback loop, and I wish there was just more political will for it and more funding for it. That's for sure. So,
Yeah.
Simon, you want anything?
No, like more botanists, more people with eyes for plants, right? That can
Yeah.
detect these things as they come in, and to be, not to misidentify plants as they think it, with the best intentions, bringing plants that they assume are relatively native or benign, but actually are not.
Mhm.
Quality control kind of thing. The San Francisco estuary, I believe, is one of, if not the most invaded estuaries on the planet. We are in a sweet spot for invasive species of all kinds, beyond plants too. And we are a port city. There are unique challenges that I hope we keep learning from and continue to grow as we figure out what being in this special place really means.
As I took a second to reflect, David, I wanted to say something else about, we don't want to run around chasing every single plant that happens to be non-native and invasive. Only 10% of the non-native plants, I believe, turn out to be invasive, something like that. So, often accused of just calling out a non-native plant. Plenty of beautiful non-native plants out there that are non-invasive. But even the ones that are invasive, we need to use data-driven tools, like some of the tools that Simon showed, and science to make good decisions as to what to prioritize. We have limited resources, we have limited time. We want to invest that in the best way possible. So chasing every single thing around is not going to get us to where we need to be. We need to make good, wise choices as to the most impactful species, the ones we can deal with in the least impactful way, and prioritize those for the most rapid response, the most rapid action, and the allocating resources towards those, rather than just chasing everything around,
Yeah.
So.
Well, thank you so much. It's very helpful. And, given how the pressures that the Bay is under, it's a very hopeful message that you bring, and hopefully it can be translated to other areas as well. So again, we thank you. And just a word about next public meeting in December. We are still working to find a speaker, and so stay tuned and watch your email, and it'll be on the website as well. So we thank you for joining us tonight, and we hope to see you next month, assuming we have a speaker, and hopefully we will. So
I would just want to say, Sandy, I see that you have a hand raised, and it also just hello. It's great to see you.
oh, I'm sorry if I missed that.
No, it's okay.
You're still muted, Sandy.
Yes, please go ahead, but you need to unmute.
You're muted, Sandy.
Sandy, yep, you're just muted.
There we go. Bingo.
I wanted to call out Eva Buxton's question about residual herbicide, because we have fought in the trenches over the need to use herbicide on these rhizominous species. So I don't want that to get neglected.
Okay.
I missed that question, and the question is residual herbicide in the substrate monitored,
Right.
and the habitat for microflora and fauna?
The very fortunate thing for us is that while imazapyr does have residual action, as we say, it can in terrestrial settings. It can actually, limit weeds from sprouting in the terrestrial settings. In the twice daily tidal in areas with the tidal flushing, we do not find that. So you see kind of the evidence of that in the photos that we've shown you of the passive revegetation that occurs in these areas, lush areas of pickleweed, other native plants, other native tidal plants, native cordgrass come into areas that have been treated with the herbicide. So these are site-specific decisions to make for using these tools, and when you want to preserve those tools when they make sense, and when they're science-driven. And in this tidal marsh system, imazapyr does not have negative impacts in that way. We have very good passive revegetation, as well as allowing us to plant directly into these areas and have them flourish as well. So thank you, Sandy, for pointing out that question.
Yes, thank you. All right, with that, and seeing no additional questions, we'll bid you good night and hope to see you next month.
Thank you very much. Thanks for
Thank you.
David. Appreciate your help.
Thank you all.